Good afternoon to all of you. Just like the other teams, we are very much an international team, and this afternoon we'll start with a French accent. So our goal was to predict Pseudo-nitzschia blooms in California. Next slide, please. The reason why Pseudo-nitzschia is important is because it's a diatom that is dominant along coastal California, reaching abundances in the millions of cells per liter. And its occurrence is usually associated with spring and summer upwelling events. Next, please. And about half of Pseudo-nitzschia species produce domoic acid, and that's a neurotoxin, meaning that it can sicken marine mammals and birds. And if we ingest it through shellfish food, then it can pose a problem for us as well, humans. So there are economic losses associated with reduced tourism on the beach and fisheries as well. Next slide, please. Dale, it's you.
I think you can keep going. I don't know if Theo is here.
Okay, so what we hope to accomplish was to make a prediction for Pseudo-nitzschia and its abundance using PACE data. And then we wanted to then compare it with a computer model called EEHolmes that is done by Coastwatch. And that forecast is trying to, again, estimate the abundance of Pseudo-nitzschia along the coast of California. And that's on the right side, for instance, you can see what one of those forecasts would look like. So you can see California, then everything is color-coded from 0 to 1 with the abundance of that diatom, in that case, over 10,000 cells per gram. Next.
Yeah,
in terms of data acquisition, sorry, go for it.
Oh, okay. Yeah, so in terms of data acquisition, we used in situ measurements of Pseudo-nitzschia cell counts from the CalHap-Map network. So we had these different stations here on the coast of California, and we used the cell counts and the domoic acid concentration data. Then we extracted the PACE data for both remote sensing reflectance, or RRS, and chlorophyll A. And we also got sea surface temperature from the NOAA gridded superset collated data set. And once we had this data, we did the 3x3 pixel average. And we did the matchups between the satellite data, the SST data, and the concentration of Pseudo-nitzschia in the water. Oh God, sorry. Okay. Our workflow, so we started by getting the data. Then we wanted to get the best predictors in terms of remote sensing reflectance. And we did that in two different ways. And then we made two different models. So the first way we did that was to create a boosted regression tree with all the available reflectance that we had to see how well it would predict Pseudo-nitzschia. And then with all of them, we selected the top three predictors, which were the reflectances at 714, 588, and 465. And then we also did a matrix of correlations. So here we have all the band ratios, and we did the division between this, and then we compared this to Pseudo-nitzschia abundances and just got the Pearson correlation to see which one would be the best ratio. Then we ran boosted regression trees with the predictors, so both the top three and the ratio. And then we made a prediction for Pseudo-nitzschia abundances for the whole region using the PACE data for the whole region. And then we compared to the CHARM model. So here are the results for the boosted regression trees. This is for the top three reflectances. So we used, as I said, 714, 588, and 465. And then we also added temperature and chlorophyll to the model. We didn't know that chlorophyll was also derived by the remote sensing reflectances, so that will probably be something we're going to remove it from the model. But nonetheless, here are the results. So we can see that the reflectance at 714, which was the top predictor, was actually the best one to explain the Pseudo-nitzschia concentrations. And here we have a plot of the predicted Pseudo-nitzschia versus the observed Pseudo-nitzschia. It's not perfect, but our person R was 0.57, and our R squared was 0.32, which we think it's pretty good as like a head start for this project. Then this is the other model that we did with the top ratio. So the top ratio we got from the correlation matrix was the reflectance at 530 divided by 532, which we thought it was a bit weird because the bands are so close, but all our matches were very close. So that's something we want to investigate why we got that result. But this is the result for that model, and we can see that it's a little worse than the other one, with an R of 0.47, and this is how the predicted versus the observed count look like. So, Bikas?
Yeah, so thank you. After we have our models trained on the sample data, we wanted to really see that if it can actually predict the bloom that we are trying to capture. So here we have selected two dates. This date corresponding to a date where we have high cell concentration. On the right hand side, we have the predictions where the yellowish regions have more cells, and if we compare that with the no probabilities, we can see that it's near the coastal regions. It's reddish, and it's more or less follows the same pattern. And yeah, we are trying to do this on a daily instance, so we may not have the data all over the place, but yeah, that's the trade-off we have to make. Next slide, please. And similar to the previous one, this is also a one-day prediction, but we have intentionally selected where we don't have much cell, or it can be also called the no bloom day. And it's same. It follows the same pattern. The no probability pattern and our model prediction shows mostly in the greenish or bluish region, which actually shows that we are actually going into the right direction. Yeah, next slide, please. Is Matthew going to talk about this slide?
Again, so one of the last slides we created is this one right here. On the left-hand side, you have the BRT prediction with the top three reflectances. In the middle is another BRT prediction using ratios only. And then we compare that to a forecast from CHARM, and that's all for August 5th, 2025. The difference right here is that we are using an 8-day map rather than a single day. We should be clear that we did not train the model on the BRT predictions is not based on anything different. So we just wanted to see if we could fill in some of the gaps right there. And what seems to be a bit strange, not strange to me, but if you look at the middle map, the BRT prediction with the 8-day, they are down at the bottom near San Diego. There is a blue blob that seems to match what we see on the CHARM model as well. Even though again that BRT the R square for that model is less, not as good as for the three reflectance. And there are a couple of blue tongues as well out in the Atlantic Pacific that also seem to match a little bit what's happening south of Monterey, for instance. So something that is a bit peculiar right here. We haven't had much time to really discuss that. Next slide, please. Bikas, is back to you.
Yeah, thank you. So for the last slide, we are just going to talk about what we can do to improve it. So within this short period, it's not possible to try out all the things. So we have to make some compromises and trade-offs. So as a future improvement, we can actually dig deep into the data exploratory analysis, and so that we can get to know our data well, where the shortcomings are and where it's performance where we can do hyperspectral signature analysis. We can also focus on features or creating more features like spectral signature and deriving more derivatives or spectral indices. We tried very simple boosted gradient trees, but there are more advanced gradient boosted models and even deep learning models that are way better, it even can perform very well. And we work with very limited data. So if we increase more samples, it might give the model more and more variability, and it can improve the model performance. Thank you.